We spend a lot of time on this channel talking about promising new remyelination possibilities, but it never ceases to amaze me that there's always more. Well, hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with MS for almost 41 years, and there has sure been a lot to cover this last couple of years on remyelination therapies that, while they're not ready for the market yet, they are edging closer and closer to it. But just because we have some that are getting fairly mature in the system doesn't mean that we're all out of ideas. Apparently, there are new possibilities coming down the pike all the time, and the mechanisms that they're looking at are different every time. So the body's very complicated, I know, but my goodness, we're really looking at some very unusual and different ways of dealing with remyelination. And I want just to cover just a couple of articles today. I apologize, I'm getting over a cold, so I may sound a little nasally or something, but I think I can still deal with all of the words and the long articles and whatnot. But we'll see what we can do, we'll see what we can find. Let's take a look. The first article that we'll look at was published in Multiple Sclerosis News Today on October 13th of 2025. It's called Two Compounds with Potential for Myelin Repair in MS Identified. Study, experimental medications showed promise in animal and cell models. Researchers have identified two experimental medications that may promote myelin repair in people with multiple sclerosis. The two compounds have shown promise in cell and animal models, and preclinical work is ongoing to bring them into clinical testing. The compounds were identified by academic researchers, but the program has now been picked up by Cadenza Bio, which will lead further development. We are committed to rapidly advancing these drug candidates with the goal of halting progression, promoting repair, and ultimately improving quality of life for patients with MS, Carol Curtis, PhD, co-founder and CEO of Cadenza, said in a company press release. The two new compounds were described in the study chlorindazole-based estrogen receptor beta ligands with favorable pharmacokinetics promote functional remyelination and visual recovery, which was published in Scientific Reports. Available MS therapies have limited effects on myelin repair, as well we know. MS is a chronic disorder caused by inflammation in the brain and spinal cord that damages the myelin sheath, a fatty coating that helps nerves send electrical signals. Myelin damage disrupts nerve communication and causes nerve cell death, ultimately leading to MS symptoms. Although there are treatments that can tamp down inflammation and slow MS progression, available therapies have limited effects on myelin repair, known as remyelination. One protein that has been identified as a potential target for promoting myelin repair in MS is the estrogen receptor beta, or ER-beta, which is produced throughout the body, enabling cells to respond to certain hormones. Data suggests that activating ER-beta in the brain can promote the production of new myelin. That's interesting. So that's yet another way that they're looking at promoting the production of myelin. With that in mind, the researchers developed and tested a number of compounds that could activate ER-beta. They ultimately identified two promising candidates called K102 and K110. A battery of tests in preclinical lab models showed that both compounds have pharmacological properties that make them well suited for use as medications. Compounds promote remyelination repair in pair of mouse models. There's those mice again. Boy, we owe them so much. In experiments done using cell models, K102 and 110 were shown to promote the growth and maturation of oligodendrocytes, the cells chiefly responsible for making and repairing myelin in the brain and spinal cord. The researchers then tested the two compounds in two mouse models of MS, one where the mice are engineered to have a myelin-targeting immune attack, and one where the mice are fed a toxin that causes myelin damage. Data from both models indicated that K102 and K110 protected myelin repair while also modulating immune responses. Again, this is quite promising, and yet we do need to keep in mind that just because it seemed to have a positive effect on mice does not mean it's going to have a positive effect on humans. But still, the preclinical work is critical to being approved to go on to the next phase. The treatments led to improvements in the health of nerve cells in the eyes, which are often damaged in MS. This is especially important because non-invasive measures of optic nerve health can be used as a proxy of myelin repair in MS patients participating in clinical trials, said Seema Tiwari-Woodruff, PhD, co-author of this study, scientific founder of Cadenza, and a professor at the University of California, Riverside. Our findings demonstrate that ER-beta ligand treatment promotes neuroprotection and enhances visual pathway function, as assessed by measures that are particularly valuable because they can also be applied non-invasively in patients, allowing direct translation into clinical trials, Tiwari-Woodruff said. Although both compounds showed promise, the researchers have identified K102 as the most promising for MS specifically. K110 is also a strong candidate, Tiwari-Woodruff said in a university news story. It has slightly different central nervous system effects and may be better suited for other conditions like spinal cord injury or traumatic brain injury, so we're keeping it in the pipeline, and good that they will, because those conditions have also been pretty difficult to deal with, and I think that the medical advances we're making in one field can definitely cross over and help us in other fields. This study was funded in large part by the National Multiple Sclerosis Society, the National Institutes of Health, and Cadenza also contributed funding for the work. And as she says in conclusion, our work represents more than a decade of collaboration, with the last 4 years focused on identifying and optimizing new drug candidates that show strong potential to treat MS and possibly other neurological diseases involving demyelination, Tiwari-Woodruff said. I found a couple of short videos related to this work. The first one I'm going to play is kind of funny. I mean, it's got good information, but the music in the background is kind of ominous almost. It's a little bombastic, but I think you'll get something out of it. So let's go ahead and have a listen.
Breaking. Potential MS nerve repair compounds, K102 and K110. If your nerves look stripped, you're losing myelin. If myelin's gone, signals slow and symptoms show. If new drugs remyelinate, hope returns for patients. Researchers discovered two small molecules, K102 and K110. They promote remyelination, rebuilding the protective sheath around nerves. They also calm harmful immune responses that damage myelin. Tested in mouse models, researchers saw improved repair, and in human cells, results matched the improvements. Both drugs improved nerve repair and restored function, but K102 stood out as the clear lead candidate. Licensed to Cadenza Bio, K102 is advancing rapidly. It is brain penetrant, able to reach the central nervous system. It's also oral friendly, suitable for pill-based dosing in humans. K102 is moving toward non-clinical work before human trials. This isn't a cure yet, but it's a major leap. It repairs myelin, restoring the nerve's protective coating, and it balances immunity, reducing harmful inflammation in one drug. If trials succeed, treatments for MS could change. And for other demyelinating injuries, the impact could be huge. Shift from managing symptoms to truly restoring function. Follow for updates. This could change everything soon.
Viviana Setola, contributor to Life News Today. Scientists from the University of California at Riverside and the University of Illinois at Urbana-Champaign have discovered two promising compounds, K102 and K110, that could repair nerve damage caused by multiple sclerosis, MS. Led by neuroscientist Seema Tiwari-Woodruff and chemist John Casanova-Bogan, the research focuses on rebuilding myelin, the protective layer that covers nerves, rather than just reducing inflammation. In early studies, K102 was able to restore myelin and normalize immune activity, both in mouse models and in human cells. The compounds originated from modifications of an older molecule, indazole chloride, and have been jointly patented by both universities. Cadenza Bio, a biotechnology company from Oklahoma, holds the exclusive license and is currently conducting safety and pharmacokinetic tests before possible clinical trials with the FDA. Funding came from the National Multiple Sclerosis Society's Fast Forward program, which helped move the project from academic discovery to clinical application. Collaborators from the Scripps Research Institute also contributed to understanding how the compounds interact with receptors related to myelin repair. If human trials confirm the current findings, K102 could become the first true remyelination therapy, one that not only stops the progression of MS, but also restores damaged nerve tissue. Researchers caution that it could be years before it reaches patients, but they see it as a major step toward moving from managing deterioration to achieving recovery. To read the complete and full article, please visit lifenewstoday.com for more information.
This next article was published in Multiple Sclerosis News Today on October 24th of 2025. It's called Protein Found to be a Natural Brake on Myelin Production in the Brain. Blocking the protein in mice led to better motor coordination. Researchers have identified a protein that naturally slows myelin production, a finding that could lead to ways to repair myelin damage in multiple sclerosis and other neurological disorders. Yeah, so in other words, repair in the sense of prevent the damage in the first place, perhaps. In mice, the protein called TFII-I, I guess, is it T5II? I'm not sure. Or T5III, I don't really know, was found to limit the activity of genes that drive myelin formation. When researchers suppressed T5II in myelin-producing cells, myelin levels increased, nerve signal conduction became faster, and the animals showed better motor coordination and other behavioral improvements. This study is among the few to identify a mechanism for increasing myelin levels in the brain. Boaz Barak, PhD, a professor at Tel Aviv University in Israel, said in a university news story. Its results may enable the development of future therapies that suppress TFII activity in myelin-producing cells to restore myelin in a wide variety of degenerative diseases in which myelin is impaired. We believe this fundamentally new approach holds great therapeutic potential, Barak said. The study appeared in Nature Communications under the title GTF2I, encoded transcription factor TFII-I regulates myelination via SOX10 and MBP regulatory elements. They talk about why scientists are focused on myelin repair for MS treatment. Myelin is a fatty coating that wraps around nerve fibers, helping electrical impulses travel quickly and efficiently. In MS, the immune system mistakenly attacks this coating, damaging both the myelin and nerve cells. This disruption slows or blocks communication within the brain and spinal cord. Myelin damage has also been associated with other neurodegenerative diseases, such as Alzheimer's disease and certain neurodevelopmental syndromes. The GTF2I gene, which encodes TFII-I, has been linked to several neurodevelopmental and psychiatric conditions, including Williams syndrome, schizophrenia, and anxiety disorders. In a recent study, researchers at TAU found that mice lacking this gene had myelin defects, but its role in myelin formation remained unclear. In the new study, researchers deleted the GTF2I gene only in myelin-producing cells in mice, leaving all other cells intact. This allowed them to test how the loss of TFII-I affected myelination and nerve function. We investigated the role of a protein called TFII-I, known for its ability to increase or decrease the expression of many genes crucial for cell function. Barak said, while TFII-I has long been linked to abnormal brain development and neurodevelopmental syndromes, its role in myelin production had not been studied until now. The results showed TFII-I normally binds to and regulates key myelin-related genes in oligodendrocytes, the cells that produce myelin in the brain and spinal cord. These genes included those encoding MBP, a central component of myelin, and SOX10, an essential protein for oligodendrocyte development. This resulted in thicker myelin sheaths around nerve fibers. The same effect was seen in Schwann cells, which create myelin for nerves outside the brain and spinal cord. Without TFII-I, these cells produced thicker myelin around the sciatic nerve, the long nerve that runs down the back of each leg. As a result, electrical impulses traveled faster across the corpus callosum, the large bundle of nerve fibers connecting the two sides of the brain. Mice without TFII-I also performed better on a Rotarod test, indicating improved balance and motor coordination, the team noted. Deleting TFII-I also appeared to increase social interactions, but led to higher anxiety-like behaviors. We found that in the absence of TFII-I, the myelin-producing cells generated higher amounts of myelin proteins, said Gilad Levy, PhD, a researcher at Tel Aviv University, and the study's first author. This resulted in abnormally thick myelin sheaths, which enhanced the conduction speed of electrical signals along the neural axons. These improvements resulted in a significant enhancement of the mice's motor abilities, including better coordination and mobility, along with other behavioral benefits, Levy added. Now, this is all they have for this article, so we are seeing that these two pieces of research are in their relatively early stages. One was referred to as preclinical, which means no human test yet. And the same thing here, they're working with mice right now. So as we can see, these are pretty early in the pipeline, but they both look pretty promising. Well, I was not able to find a good video about the second research that we've been looking at, but I did find a website for Boaz Barak's lab, the Brain Behavior Lab in Tel Aviv University, I assume. And the website is titled Exploring Myelination and Brain Development in Neurodevelopmental Disorders. And there's a lot of information on here, as you can see, publications that they've had recently, and different interviews that have been done, not on this research, but on other related research, and a selected talk even on glutamate signaling remyelination in autism. So that might be of interest to you, but I will link this site below. The other thing they have here is a way to stay updated with their research by subscribing through email, which I did do. So if there's anything new that they tell us about this new and latest discovery related to MS, I will be sure to let you know. Now again, this one is just looking at mice. We aren't ready for people yet, but interesting too, because it's coming at it from the opposite side of things. It's looking at ways there are mechanisms in the body that actually block the production of myelin. Who knew? It has to do with the oligodendrocyte precursor cells, because if they don't mature, then they don't become the types of cells that can make myelin. And if anything gets in the way of that maturation process, we did look at something pretty similar before. So they're looking into that more. Let's see what they find. And I will, again, definitely keep you posted about that. There are an endless number of teams out there looking at endless variations on this theme. So I have a feeling I could do just an entire channel solely devoted to remyelination. I don't know that that would be the end of the world, because to me, even though, like I said before, I'm probably a little too advanced in my course of MS to gain any real benefit from it, it's the hope for the future of MS, I think, unless we can eradicate MS altogether from the population, which would be great. But if we can't, I think remyelination is the very best way to keep people symptom-free and living the quality of life that they really want to live. I'd be interested to know from you if either of these two things sound plausible. I know it's frustrating that they're so early in the pipeline, but even if they were ready and available for us right now, would that be something that you would be interested in getting involved in, or a clinical trial, or even if your doctor said it was ready for you to try, would you try it? Let me know. I would really love to hear from you. But that is all I have for you today. So until my next video, please do take really good care of yourself, and I will see you again soon.